First of all, I would like to acknowledge the organization for the opportunity to be here to present the work that we have been developing in the Tribis Research Group in the University of Minho in Portugal about 3D printing of skin substitutes. So one of the technologies that we have been exploring mostly in the group is called cell sheet engineering, and this technology relies on the capacity cells have to deposit their own matrix and then to form cell sheets that can be then stacked to form tissue-like substitutes. So this is a technology that has as main advantage to be completely biological, so it's a biomaterial-free technology, and also has the importance of allowing the preservation of cell-cell and cell extracellular matrix interactions. On the other side, each cell sheet is a 2D system, so we only have 3D tissue substitutes when we stack the different cell sheets. We have the limitation of be able to use this technology only with cells that deposit a significant amount of extracellular matrix, and within each cell sheet there is a limited cell organization. So, in opposition to this technology, we have been also using 3D printing, and everybody knows that the major advantage of this technology is to be able to tailor the distribution of the different cells and materials within each layer, to be able also to generate large 3D structures. However, there's still a major limitation in the field regarding 3D bioprinting in particular, that is related with the bioinks that have been developed so far, and that should be tailored to the specificities of the different tissues that we want to. Gel gum is one of the materials that we have been exploring mostly in the group, not only as part of inks or bioinks, but also for many other biomedical applications. And this polysaccharide has the advantage of forming gels by temperature decrease, and also to further reinforce the network that it forms after interaction with cations. So these are characteristics that are quite interesting for 3D printing. But it's also important to understand at which temperatures these networks of gel and gum can be formed, and you can see that the range of temperature is between 25 and 37 degrees C, so they are quite compatible with the printing of cells without causing any harmful effects. In addition to these characteristics, it's also important to understand and to see that gel and gum solutions also have a shear thinning nature, which means that when they are subjected to stress, their viscosity decreases. Again, if you think on printing, you can see that this decrease in viscosity is beneficial for not causing any additional stress to the cells that are being printed. So gel and gum is actually a very versatile biomaterial, and as I said, we have been exploring also for many other different applications in the biomedical field. And in this particular work, we were able to modify gel and gum with this divinyl sulfone group, which allows us to have a material to which virtually we can link any peptide that we are interested of, as long as they have a cysteine residue to link to this moiety. So when we think on a bioink, we can have actually the same base of gel and gum modified with the peptide of interest that also links to one kind of cell or different kinds of cells, depending what is our purpose, in order to be able to build complex 3D structures. When we first started using these gel and gum based bioinks and modified with peptides, we looked at the epidermis and then to the basement membrane that is composed of collagen 4. And then we select this peptide, HEP3 peptide, that is a sequence that is present in the collagen 4, and combined with the modified gel and gum at different percentages. And you can see here, this is just an example of the fibers that were printed with these materials. And here, when the keratinocytes, the epidermis cells, were encapsulated or incorporated in these bioinks, they actually had a very similar behavior independently of the polymer amount. They were actually round and adhered to the material, and they were not very influenced by the polymer concentration. When we looked at the vascularized dermis, and here you looked at the two different components, biological components, the fibroblasts that are the main cell type in the dermis, and the endothelial cells, microvascular endothelial cells in this case, to look at the vascularization, we could actually see that in opposition to the keratinocytes, the fibroblasts had a preferred formulation of the material in which they adhered better, and they actually proliferated and deposit matrix that is typical of the dermal part. This is interesting to see that when we looked at the endothelial cells, and this is the three images in the bottom, in fact, we have a better behavior, or it is also compatible with the same formulation that we use for the fibroblasts. So we are able to create a dermis substitute using this formulation, because both cell types that are of interest behave better in addition to the main skin parts, the epidermis and the dermis, we have been also looking to the appendages of the skin. And in this particular case, I would like to show you some of our results for the hair follicle. And in terms of rationale, we had exactly the same as using the Gel-N-Gan modified with different peptides. And in this case, we looked at the dermapapilla, that is the inductive part of the hair follicle, and also the presence of the collagen 4, and we select also the EP3 peptide, as I showed before, for the epidermis, and also in the major component fibronectin, and we selected this RGD sequence to link to the Gel-N-Gan. So if we look here again, the same analysis, this is for the RGD peptide, and this is for EP3 peptide, and again the dependence with the polymer concentration, we can clearly see that this is the addition for both EP3 and RGD are dependent on the polymer concentration or amount, and it's very clear that EP3 they are able to adhere in a more extent way to the EP3 peptide. If we looked to the production of matrix, we can clearly see that, like for the fibroblasts, for example, you also have the production of key extracellular matrix components, but most importantly, we have this ALP activity that is quite strong, and this is a marker that is typical of dermapapilla cells that and that shows their or represent their inductive behavior. And if we look also to the amount of ALP, as well as to the general quantification of collagenoses and non-collagenoses proteins, we can clearly see that the percentage of the polymer is, in the higher percentage, is beneficial for this for the culture of the dermapapilla cells. And interestingly, you can see that there's no significant differences in terms of ALP, and by itself the 3D environment is also relevant to restore this activity of ALP activity. But we have a specific response to EP3 and RGD peptides in terms of the type of proteins that are deposited in these inks. As highlighted for the cell sheet engineering technology, extracellular matrix is a key part of the replication and the construction of tissue-like substitutes. because extracellular matrix is not only a mechanical support and provides integrity, but it's also a reservoir or biochemical cues. And it also influences, of course, and the first impact is the way cells see it immediately, and when you consider the bioink immediately after the deposition of the bioink and the construction of the 3D construct. So when I talked about the different biomaterials, we had in consideration the type of proteins that are present, and we mimic that somehow with the peptide sequence that we included in the gel and gum material. But so far, no natural or synthetic material can fully replicate all the features of the natural extracellular matrix. So having this in mind, and in a different work, this is just to show you how important this is and how relevant it is for us to look at the extracellular matrix. We could see that, for example, this is for epidermolysis bullosa, and you can see from the analysis that we did the different variants of the disease. Clearly, cells have a different profile of the extracellular matrix that is produced, and of course, in the end, this is reflected in the different processes, biological processes in which they are involved, and in the end, that represent or are traduced in the pathophysiology of the disease. So we've been also looking at from this perspective and to try to develop bioinks prepared from extracellular matrix produced by different cell types. And here you can see that we developed a methodology to extract this ECM, and so far we have been able to actually have recovered a significant amount of the content of the extracellular matrix in terms of protein and glycosaminoglycans. And we have also been able to see that we have different particles that form this extracellular matrix that we extract, that have a different range of sizes that we can play and use for different purposes. In addition to this extract, we have been also able to obtain a second extract from the same native extracellular matrix. And interestingly, when we looked and when we did a proteomic analysis for those, we could see that they actually complement each other very nicely, and we have very important biological functions that are associated to the components that are present in these extracts. So we have been using these extracts in combination again with gel and gum to obtain different inks and to be able to use them to build tissue-like substitutes. So far, you can see here that they extrude nicely. This is just a needle, but it could be the needle of a printer. We have been trying and looking at an array of different percentages of matrix and gel and gum to check on the stability. And we could see that even after 7 days, the amount of protein that is retained within the gels is significant, is almost the same as the one that we incorporated, which means that they form actually stable structures with the gel and gum. You can see here, this is a CoLF staining that shows the matrix that we incorporated together with the gel and gum. This is the control. When, of course, we did the rheological analysis and all the analysis to be able to understand if these mixtures that we have been producing are suitable for 3D printing, we could actually see, again, we have been varying, as I told you, the different percentages of the components. You can see that some of them are actually in a range of temperature that will allow to print together with the cells without expecting having significant negative effects in the cells. We could also see that, again, they have this shear thinning behavior, so the viscosity diminishes with increasing stress, except for this material that is just our control, the gel and gum, with different percentages of very low percentages of polymer. And importantly, we did a test that shows that after printing, for example, we could actually, the material recovers their properties, so it allows actually to take advantage of the wall characteristics that it presents. We also did some preliminary tests with cells, and you can see that there's in fact an effect. We have cells that are linking to the extracellular matrix components in opposition to the material without this component, and are able to proliferate. So just take home messages, and to conclude, it's still to be defined the ideal bioink formulation. So it's still needed to be understood if we can have biomaterials that replicate sufficiently the native extracellular matrix. For skin regeneration and potentially for other tissues, we still need to go a little bit further and to promote tissue maturation and to understand to what extent remodeling happens in our 3D printing tissue substitutes. And if we think on skin tissue models that can be also used for generating basic knowledge on wound healing, or for testing improved therapies with higher correlation to humans than animals, we actually really need to consider that we have to have a better mimic of the extracellular matrix in those models to actually be able to have representative results. So just to finish, I would like to acknowledge the funding, the international collaborators, also the national collaborators, particularly the clinicians, as well as the team of people that was involved in developing this work. Thank you very much.